this lecture, we're going to talk about the role of immunological memory in protecting individuals from second or third infections from the same pathogen. We're then going to apply these principles to understand how does vaccination work. Now, you may recall from our past lectures that B cells and T cells will generate memory B cells and T cells. And so we talked about how when T helper cells are stimulated, we can produce memory T helper cells, and that similarly also happens with TC cells and B cells. And this is part of that response to cytokines. Well, the whole role of this then, or the purpose of memory, is so that an immune system will be able to recognize a pathogen sooner and respond to it more quickly the second time around. And note here again that the T helper cells and the TC cells make up our cellular immunity, whereas the B cells make up our humoral immunity. And particularly the role of those B cells is to secrete antibody. And so if we look at the graph on the right-hand side here, we're looking at the concentration of antibody over time. Well, upon the primary exposure, or the first exposure to some pathogen or epitope, what we find is that there is a smaller production of antibody than we see during a secondary exposure or secondary response. And so the point that is really to be taken away here is that our response to the second or third exposure to an antigen or an epitope is oftentimes much stronger than the first time that we see this. And this again is because of the adaptive immune system. Now, when we talk about the immune response, we oftentimes can classify immunity into 4 different categories. We can talk about naturally acquired active immunity, and this occurs when somebody is infected with a wild type strain of some pathogen in our body, generates an immune response to it. We can talk about artificially acquired active immunity. This occurs in the case of a vaccination. And then we can also talk about passive immunity, in which somebody is receiving antibodies from somebody else. Really, what we say here is that in active immunity, my immune system or your immune system is generating a response, producing antibodies, and producing memory B and T cells. However, in passive immunity, we're receiving immune response, or typically antibodies, from somebody else. For example, in naturally acquired passive immunity, what we see here is a baby receiving antibodies through mother's milk, and this is actually quite common. Because babies have a naive immune system during the first 6 months, they receive most of their antibodies from mother's milk. In artificially acquired passive immunity, this would be the use of antibodies or an antitoxin to prevent a toxic shock or toxic disease within an individual. Now, the goal of vaccination is really to stimulate one's own immune response to generate memory cells. We saw in the previous picture how a secondary exposure to an antigen leads to a much stronger response of antibody production. And the goal of vaccination then is to basically train or to give the immune system a round of practice to generate these antibodies before the individual is exposed to a natural form of the pathogen. For the rest of this talk, we are going to focus on artificially acquired active immunity and vaccination and the role of vaccination in generating these memory cells. Now, the goal of vaccination, again, is to generate memory cells that will be able to recognize a pathogen when our bodies see a wild type or natural strain of a pathogen. However, vaccines don't only protect those who receive the vaccine, they also protect those who are immunocompromised or who cannot get the vaccine. This concept is known as herd immunity. And if you think of a herd of elephants, for example, in a herd of elephants, the elderly and the young are put towards the center and protected by the more strong elephants on the outside. Well, this is the same concept that we talk about with vaccination. We have this protect the herd type mentality. If we vaccinate those who are immunologically strong or who have good immune systems, then we can protect those who are immunologically weak. Take a look at the examples on the right here. In the first example, we have a population that is naive to a particular type of pathogen. Blue indicates that they're not immunized but healthy. Red indicates that they're not immunized and sick. And yellow indicates that they're immunized or that they have been vaccinated or previously exposed to the pathogen and so are unlikely to get the disease a second time. Well, in a population that is immunologically naive, one that has not seen the pathogen yet, what we find is that these diseases can spread quite rapidly. This is the type of case that we see with diseases like Zika, Ebola, and with coronavirus, where we don't have vaccines currently available, and these diseases can spread very easily from one individual to the next. Next, our second example down shows a population where maybe some people have been exposed to the pathogen and others have not, and yet still in this case here, we don't have enough of a population that has been exposed to the pathogen to protect the herd. But in the last example, what we find is a very high proportion of individuals that have been immunized. And again, this immunization can either occur naturally or it can occur artificially through the direct exposure to an antigen of the pathogen. And in this case here, what we find is that the population of individuals who have been immunized protect those individuals who have not been immunized and still remain healthy. Now, what we're really saying here is that a certain percentage or a certain threshold of the population must be vaccinated in order for herd immunity to be achieved. Obviously, in our three examples on the right-hand side here, the top two show cases in which herd immunity has not been achieved. The percentage of individuals that are previously exposed and immunized against this particular pathogen is not great enough to prevent its spread. However, we do see that herd immunity threshold has been achieved in the last example. Now, it's important to note that there are a number of factors that can influence what this percentage of individuals is. And those factors can include things like the mechanism of how the disease spreads, how many individuals are infected from every one individual that is currently infected, something that we call the R sub naught value or R0 value. And genetics can also influence this herd immunity threshold. Just to give you some context for the size of these thresholds, for measles, it's believed that the herd immunity threshold is 90%. In other words, 90% of the population must be vaccinated or previously exposed to measles in order to protect the herd from community outbreaks. Now, what we're going to do is we're going to cover 4 types of vaccines. The first we're going to talk about is an attenuated vaccine, which is sometimes called a live vaccine. Really, though, what's important to note here is that this is a weakened form of a pathogen. And so it may cause mild symptoms and disease, but it really mimics the natural infection. And because of this, because of the ability of this weakened form to be able to replicate inside the host, it actually stimulates T helper cells, B cells, and those cytotoxic T cells. And that really makes these attenuated vaccines, if they can be created, some of the best vaccines out there, although they run the risk of not being safe, particularly individuals who are immunocompromised, for example, HIV positive patients, individuals who have cancer or maybe have a transplant and so are put on immunosuppressive drugs. These people should not receive these vaccines because the symptoms and disease may be more severe in their case. Let me give you a quick case study on some of the attenuated vaccines. One of the original vaccines for polio is known as the oral polio vaccine, or the OPV. This is an attenuated vaccine, and it worked incredibly well to reduce the number of cases and incidences of polio. However, we're at a point now where the number of infections in some years that occur naturally from polio is lower than the number of infections that occur from the vaccine. And what that points out here is that these attenuated vaccines sometimes may revert to more pathogenic forms or more virulent forms that cause disease. And so we have to be cautious when using these attenuated vaccines. We'll talk towards the end about a different type of vaccine that would not have the negative side effects, but would still have the same sort of upside as these attenuated vaccines do. Now, on the other hand, inactivated vaccines, or what we sometimes call dead vaccines, are safer. These are treated forms of pathogen that can no longer cause disease. Treatment can involve heat or some sort of chemical treatment, like the use of formaldehyde to prevent proteins from functioning anymore. The downside of these inactivated vaccines is that they, and you could view this as an upside as well, I suppose, is that they don't replicate in the host, and they don't cause disease or symptoms. And so these ones would be safe to use in an immunocompromised individual. However, because the vaccine, or because the pathogen and its antigenic material cannot be produced or replicated inside the host, then there's not going to be any presentation of epitopes on MHC1. This limits the memory of the immune system to inactivated vaccines. While memory B cells and T helper cells will be produced, memory TC cells will not be produced. And oftentimes we think that these inactivated vaccines may require booster shots or booster vaccines down the road. In general, we believe, or evidence suggests, that inactivated vaccines do not produce as robust an immune response and as much long-term memory as we find with activated or attenuated vaccines. Subunit vaccines are our next class. These are vaccines made up of an antigen, or oftentimes just one part of a pathogen. And often this part is something that is strongly immunogenic, or something that triggers a strong immune response, like a protein. Because this is just a single protein, these vaccines will not cause disease. They also don't reproduce in the host. And so, again, because of that, these stimulate a similar response to those inactivated vaccines. They will stimulate the production of memory T helper and memory B cells. Now, it took a while to get to the point where we could generate subunit vaccines. Actually, it required us to understand biotechnology and to develop that field. And so these types of vaccines have only been around maybe for about 30 years or so. Oftentimes what happens here is the protein is encoded by a gene that is produced in a microbe like E. coli or yeast in some case, and then that protein is purified. And after the purification process, it is then ready for vaccination. The Gardasil vaccine, or the HPV vaccines that exist right now, are ones that are subunit vaccines. In particular, those vaccines rely on the use of the protein that makes up the viral capsid to generate what we call viral-like particles. These little particles have the kind of spherical shape of the HPV, but do not contain any genetic material, and so they cannot replicate in the host. Now, there are two subclasses of subunit vaccines known as toxoid and conjugate, and we're not going to get into the details of these, but if you do see these two terms, know that these are both types of subunit vaccines. Now, there's been a lot of work done on vaccines over the past 20 years or so, and one of the new avenues that's still being explored is the use of what we call nucleic acid vaccines. Nucleic acid vaccines would use DNA or RNA, and that would be injected into an individual. The idea here is that that DNA or RNA would make its way into cells. In the case of DNA, it would be transcribed, and then the mRNA would be translated into a protein that is an antigen for some pathogen. Now, the benefit of this, it looks a lot like, or it kind of functions like a subunit vaccine in that only one protein is being produced. And again, this would not cause disease or symptoms of disease in an individual. So theoretically, these nucleic acid vaccines would be safe for an immunocompromised individual. However, because it would be our own cells and our own machinery that is producing the proteins for this, that would mean that these antigens could be presented not only on MHC2, but also on MHC1. And that makes the potential for these nucleic acid vaccines to be much greater than what we've seen with subunit vaccines. In fact, research suggests in mouse models that these types of vaccines could stimulate the production of memory T helper cells, T cytotoxic cells, and B cells. Currently, nucleic acid vaccines are being looked at for not only to generate vaccines for HIV, but for also the novel coronavirus, which caused a worldwide pandemic in 2020. All right, so in summary, I think there are really three main takeaway points here. First, vaccination is the intentional exposure of an individual to an antigen, and the goal of it is to generate immunological memory. It's to train the immune system. This is a form of artificially acquired active immunity. Secondly, vaccination protects herds. In other words, it's a form of community protection against disease or illness. And finally, different types of vaccines have their individual strengths and weaknesses. While some will stimulate T helper, TC, and B cells, and some do not, the drawback of some may be that they cause illness. And so there really is a balance between here how strong of an immune response does it generate, and what is the risk we're willing to take in order to generate that strong immune response.